Good morning, students. Welcome back to Open Web-based learning platform of St. Joseph College for Women, Visakhapatnam. Today we are going to start with another topic called Equisetum, which is known as horsetail. If you see the systematic position of the Equisetum, it is it belongs to division Pteridophyta. Class is Sphenopsida, order is Equisetaes, family is Equisetaceae, and the genus is Equisetum. It is the living fossil with the primitive feature. This is the morphological nature of the Equisetum, the horsetail. If you see the habit and habitat of the Equisetum, plant body of Equisetum has aerial part and underground rhizome part. The rhizome is perennial, horizontal, branched and creeping in nature. Aerial part is herbaceous and usually annual. The majority of the species are small with a height of range 15 to 60 centimeter and a diameter of 2 centimeter. Some species of Equisetum giganteum are up 13 meter while Equisetum telematia is 3 meters and Ramosisolium is 4 meters. Though there are stems in a relatively thin of 0.5 to 2 centimeter in diameter, showing vein-like habit and climbing over the adjacent forest trees. Equestrians generally grow in wet or damp habitats and are particularly common along the banks of the stream or irrigation canals. Some species are adapted to xeric conditions like Equisetum arvens. Common Indian species are Equisetum arvens, Equisetum debile, Equisetum diffusum, and Equisetum ramosimum. Some species of Equisetum are indicators of the mineral content. Some species accumulate gold, about 4 to 5 ounce per ton of dry weight. They are considered to be as gold indicator plants. Hence, these plants helps in prospection or exploration of new ore deposits. In Equisetum, silica is deposited in the older walls of the epidermal cells, giving the characteristics rough fillings. Thus, it provides a protective covering against predators and pathogens. If you see the structure of the equisetum, the sporophytic plant body of equisetum is differentiated into stem, root, and leaves. The stem has two parts, the perennial underground much branched rhizome and an erect, usually annual aerial shoot. The branching is monopodial. Shoots are differentiated into nodes and internodes. Majority of the species, all the shoots are like and chlorophyllous, and some of them bear the strobilus at their apices. In case of Equisetum ramosimum and Equisetum debile. Sometimes shoots show dimorphism, vegetative and fertile. Some shoots are profusely branched, green, and purely vegetative. The others are fertile, unbranched, brownish in color, and have terminal strobili. The underground rhizome and the aerial axis appear to be jointed due to the presence of distinct nodes and internodes. Externally, internodes have longitudinal ridges and furrows, and internally they are hollow tube-like structures. The ridges of the successive internodes alternate with each other, and the leaves are normally of the same number as the nodes. Internal, if you see the anatomical features of the stem, the tiers of the stem appears wavy in outlets with ridges and furrows. Epidermal cell walls are thick, cuticularized, and depositions of the silicous material. Stomata are distributed only in the furrows between the ridges, and a hypodermal sclerenchymatous zone is present below each ridge and may extend up to the stele in gigantium. The cortex is differentiated into outer and inner regions. Outer cortex is chlorhynchimatous while inner cortex is thin-walled parenchymatous cells. There are large air cavities in the inner cortex corresponding to each furrow and alternating with the ridges known as vallicular canals. They are Cyagenous canals extending the entire length of internodes and form a distinct aerating system. Stele is ectoploic syphnostic, which is surrounded by outer endodermal layer, and inner endodermis is also present in some species of the Equisetum. The endodermis is followed by single layer of pericycle. Vascular bundles are arranged in a ring that lies opposite to the ridges in position and alternate with the valvular canals of the cortex. Vascular bundles are conjoined, collateral, and closed. In mature vascular bundles, protoxylem disorganized to form the carinal cavity, which lies opposite to the ridges. So this is the TS of the Equisetum stem, where you see the ridge by wavy like or section outlines, where it has a ridge and furrows, and the epidermis is single layered, followed by with outer by having the cuticle. Beneath the epidermis is the cortex, where the cortex is two regions, the outer cortex and the inner cortex. The inner outer cortex is thick, and the inner cortex thick wall, and inner cortex is thin wall made up of parenchymatous. At the furrows, you will be finding the canal cells, which we know the vallicular canals, and then inner is the limiting membrane cortex is known as the endodermis, and in local is internally is present the stele, and the stele is what is known as the ectoploic syphostele. You can see the stele having at the furrow ridges you will be having what the cranial canals. So the xylem is so the vascular bundles that are present are conjoined, collateral, and close, where the xylem and the phloem are present. The protoxylem cells will form a cavity-like structures, which we call it as the cranial canal cell. In the center is present the pith. The internal structure of a shoot of Equisetum shows xerophytic as well as hydrophytic feature. The xerophytic features are the ridges and furrows in the plant, deposition of silica in the epidermis cells, the sunken stomata, sclerenchymatous hyperodermis, reduced and scaly leaves, and photosynthetic tissues in the stem are the xerophytic adaptations. The hydrophytic adaptations are well developed alternating systems like carinal canals, valvular canals, and central pith cavity, and reduced vascular elements. If you see the root, the primary root is ephemeral. The cylinder adventitious roots arise endogenously at the nodes of the stem. The tiers of the root show epidermis, cortex, and the stele. Epidermis is consists of elongated cells with or without the root hairs. The cortex is extensive. Cells of the outer cortex have thick walls, sclerenchyma, and those of the inner are thinner parenchymatous. Stele is protostelic, where the xylem is triarch or tetrarch, or smaller root where may be diarch. The large metaxylem element is present in the center, and the stele protoxylem stands light around it. The space between the protoxylem groups is filled with phloem, and there is no pith. So this is the structure of the root, where you will be finding the outer epidermis with the root hairs, inner the cortical cells, the outer and inner cortex. Inside the cortex are present the stellar region, where the stele is protostele with the diarch condition, metaxylem in the center, and the protoxylems towards the periphery. The leaves are small, simple, scale-like, isophilous, attached at each node, united at least a part of the length, and thus form the sheath around the stem. Sheath has free and pointed teeth-like tips. Number of leaves per node varies. The number of leaves at nodes correspond to the number of ridges on the internode below. The leaves do not perform any photosynthetic function, but they provide protection to the young buds at the nodes. So this is the structure of the where you will see outer epidermis, then the cuticle. Inside the cortex is having the parenchymatous cells with chlorophyll cells, chlorophyll present in that, known as the mesophyll cells, and inner is the vascular bundle, where you will be seeing the xylem in the center surrounded by the phloem. Regarding the reproduction in Equisetum, it takes place by vegetative, asexual reproduction by spores, and then bisexual reproduction. Vegetative reproduction takes place by means of rhizomes, which are subterranean in some species, and for which forms the tubers, which on separation form the parent plant, which germinate to produce the new saprophytic plant. The tubers develop due to irregular growth of some buds at the nodes of the rhizome. S- Then the second type of the reproduction is by means of spores. These are produced within the sporangia. The sporangia are born on the sporangiophores, which are aggregated into compact structure, which termed the strobilus or cone or sporangiophore spikes. Strobilus is terminal in position and is born terminally on the chlorophyllous vegetative shoots. Strobilus is composed of a central axis with whorls of sporangiophores. They are stalk structures bearing hexagonal peltate disc at a distal end. On the surface of the sporangiophore, 5 to 10 elongate cylindrical hanging sporangia are born near the periphery inner ring. If you see the structure of the meiosporangium, it is elongated sac-like structure attached to the inner side of the peltate disc of sporangiophore, surrounded by jacket layers, which is composed of two layers of cell. Inner layer is generally composed of the cells of the outer layer, have helical thickenings which are involved in sporangial dehiscence. At maturity, the strobilus axis elongate. At its result, the sporangiophores become separated and exposed. Then the sporangium splits open by longitudinal line due to the differential hygroscopic response of the cell walls. The spores are spherical and filled with densely packed chloroplast. Spore wall is laminated and the spores uh, shows four concentrated layers. Innermost is delicate intine followed by thick exine and middle cuticular layers and the outermost epispore or the perispore. Intine and exine are the true walls of the spore. At maturity, the epispore splits to produce four ribbon-like bands and the strips of the flat spoon-like tips. The bands are free from the spore wall for the common point of attachment and remain tightly coiled around the spore wall until the sporangium is fully matured. They are called elaters. These elaters are hygroscopic and are spirally coiled around the spores. The spores dry out at maturity and consequently the elaters becomes uncoiled. The quisetum is homosporous pteridophytes. The haploid spore germinate to form the gametophyte. Germination takes place immediately if the spores land on the suitable substratum. The spores swell up by absorbing water and shed their exine. First division of the spore results in two unequal cells, a small cell and a large cell. Smaller cells elongate and form the first rhizoid. The larger cells forms the prothallus, and the prothallus bears the sex organs and reproduce by means of sexual methods. So these are the different the spores, which is multiflagellate. These are the elaters, and the spores. These develop into the prothallus. The exine is ruptured, the entire they form to two cells. The lower cell forms the rhizoid, the upper cell forms the base which forms the prothallus, and inside are present embedded the sporangia, embedded the gametophytes. So if you see the sexuality in Equisetum, the gametophytic plant body, which is known as prothallus, bears the sex organs, the Antheridium and Archegonia. The gametophytes are basically bisexual, homothallic that bear both male and female sex organs. Although some unisexual members are also reported, some are initially unisexual and then become bisexual. In monoecious species, Anthria develop later after Archegonia. There are two types, projecting type and the embedded type. The periclinal divisions of the superior antheridial initial give rise to jacket initial and androgonial cell. Each jacket initial divides anticline to form the single layer jacket. The repeated divisions of androgonial cells form numerous cells which on metamorphosis produce permitted anthrozoid. The anthrozoids escape through the pores created by separation of the apical jacket cells. Each anthrozoid has about 120 flagella attached to the anterior end. So this is the antherozoids, where from the antheridial initial they form the jacket initial and androgonial initial. The jacket initial go anticlinal division and form a single layer of jacket layer. The androgonial cells divide and forms the multiflagellated spermatozoids. The second, the other is the female gametophyte, which we call it as Archegonium. Any superficial cell in the marginal meristems acts as archegonial initial, which undergoes periclinial division to form a primary cover cell and inner central cell. The inner cover cell, by two vertical divisions at right angle to each other, forms the neck. The central cell divide transversely to form the primary neck and the venter cell. The maturity archegonium has a projecting neck comprising three or four tiers of neck cells arranged in four rows to neck canal cells of an equal size and a ventral canal cell and egg at the base of the embedded venter. The archegonium is confined to cushion region in between the aerial lobes. So you can, this is the archegonium with the neck cell, the neck canal cells, the venter canal cell, and the egg cell. Fertilization, water is essential. The gametophyte is be covered with thin layer of water in which the antherozoids swim to the archegonia. The neck canal cell and the venter canal cells of the archegonia disintegrate to form a passage for the entry of antherozoids. Many antherozoids pass through the neck canal cell and of the archegonium. Only one of them fuses with the egg. A diploid zygote is formed and more than one archegonia are fertilized in a prothallus. The embryo is the new sporophyte, and the embryo is the mother cell of the next sporophytic generation. First division of the zygote is transfer, result into a upper epibasal and lower hypobasal. Embryo is therefore exosporic, where the apical cell is directed outward towards the neck of the archegonium in polarity. No suspensor is formed in Equisetum. The epibasal and hypobasal cells divide at right angles to the ogonial wall and result to the four-cell quadrant stage. All the four cells of the quadrant are different size and shape. The four-celled embryo undergoes subsequent division, and the future shoot apex originate from the larger cell, and the leaf initials from the remaining cells of the one quadrant of the epibasal hemisphere. So this is the development of the sporophyte from the embryo. And if you see the life cycle in horse tails, you find the alternation of generation where the dominant phase is the sporophytic phase and the gametophyte produce the male and female gametes. Fertilization leads to new sporophyte and the alternation of generation is the heteromorphic type of alternation of generation. Regarding the economic importance of horsetail, it is used as a scoring material due to silica. It is used in traditional systems. It is a soil stabilization and it is a indicator of moist habitats. So Equisetum is a unique pteridophyte, shows primitive and advanced features important for the evolutionary studies. Thank you.